Nonspecific body defenses, which are also known as innate defenses. The first line of defense are surface membrane barriers. These include skin and mucous membranes. Skin serves as a physical barrier to pathogens as well as a chemical barrier. The sebum in your skin has a lower pH level, about 5, which inhibits growth of pathogens and encourages the normal growth of flora. The mucous membranes include eyes, nose, and respiratory tracts, GI, and GU tract. The eyes use tears to wash away any microorganisms. They also contain lysosomes, which is an enzyme that kills bacteria. The nose is full of hairs that trap dust and dirt. Cilia is found in your respiratory tract that moves mucus out. GI tract has a boiling pit of acid of pH 2 that kills some bacteria but not all of it. GU tract differs between males and females. The males have a longer urethra and contain a more acidic environment, which because of this they are less likely to have problems with UTIs. Females, on the other hand, are more prone to infection because they have a shorter urethra, even though their vagina has an acidic environment. If a pathogen gets past the first barriers, your nonspecific cellular and chemical defenses are your second line of defense. These act as the same strength every time and are not specific to a pathogen. These include phagocytes, natural killer cells, inflammation, and antimicrobial proteins. Phagocytes include macrophages and neutrophils. Macrophages are found in chronic sites of infection. There are two different types. The first is called a free macrophage, which wanders freely throughout the body. The second type is a fixed macrophage, which does not go anywhere. An example would be the Kupffer cells in your liver. Another important thing to know about macrophages is that when they are in your bloodstream, they are called monocytes. The monocytes develop into macrophages when they leave the bloodstream and enter the tissues. The other type of phagocyte are neutrophils, which are found in the site of acute infections. These are the most abundant of white blood cell. Neutrophils phagocyze infections and destroy themselves in the process. One of the tactics of neutrophils is their release of defensins, which is a substance that contains little spears that poke holes in bacteria. Phagocytosis is the process in which a phagocyte engulfs bacteria or a pathogen and ultimately destroys it. The process starts with engulfing a pathogen, which then forms a phagosome, which is a vacuole that fuses with a lysosome. If the pathogen is resistant to the acid hydrolysis of the lysosome, then helper T cells, we will get there later, release chemicals that stimulate the macrophage, which activates additional enzymes that produce a lethal respiratory burst. Another way of our body destroying pathogens is the process of opsonization, which is the process of adding carbohydrate handles to the outside of the pathogen, making it easier to kill and speed up phagocytosis. Natural killer cells police the body and lymph. There are a unique group of defensive cells that can lyse and kill cancer cells and virus-infected body cells before the adaptive immune system is activated. NK cells are a part of a small group of large granular lymphocytes. They also promote an inflammatory response. Speaking of inflammatory response, let's talk about inflammation. The steps of tissue response to injury. Inflammation is triggered whenever body tissues are injured by physical trauma, intense heat, irritating chemicals, or infection by viruses, fungi, or bacteria. Some inflammation chemicals include histamine, which causes inflammation, kinins or kinins, which causes pain and inflammation, prostaglandins, which cause pain and inflammation, complement, which causes inflammation, and lymphokines, which is a chemical signal. Actions that take place in the inflammation process are vasodilation and increased vascular permeability. Vasodilation accounts for two of the cardinal signals of inflammation. The redness and the heat of an inflamed region are both due to local hyperemia. Local hyperemia, which is congestion with blood, that occurs when local arterioles dilate. Inflammatory chemicals also increase the permeability of local capillaries. Soon after inflammation begins, phagocytes flood the damaged area. Neutrophils lead, followed by macrophages. If pathogens provoked the inflammation, a group of plasma proteins known as complement is activated, and elements of adaptive immunity, which are lymphocytes and antibodies, also arrive at the injured site. During phagocytized mobilization, injured cells release chemicals called leukocytosis inducing factors that are going to release neutrophils. Positive chemotaxis are inflammatory chemicals that act as a homing device or are more precisely chemotactic agents. Neutrophils and other white blood cells migrate up the gradient of chemotactic agents to the site of injury. Margination are inflamed endothelial cells that sprout cell adhesion molecules known as CAMs that signal this is the place. Margination refers to the phenomenon of phagocytes clinging to the inner walls, which are the margins, of the capillaries and post-capillary venules. Diapedesis, our continued chemical signaling, prompts the neutrophils to flatten and squeeze between the endothelial cells of the capillary walls. Antimicrobial proteins are a group of proteins that attack foreign invaders, and they are nonspecific. A variety of antimicrobial proteins enhance our innate defenses by attacking microorganisms directly or by hindering their ability to reproduce. The most important antimicrobial proteins are interferons and complement proteins. Fever is an abnormally high body temperature, which is a systemic response to invading microorganisms. When leukocytes and macrophages are exposed to foreign substances in the body, they release chemicals called pyrogens. These pyrogens act on the body's thermostat and reset your body's thermostat. Fever is an adaptive response that seems to benefit the body, and additionally, fever increases the metabolic rate of tissues and may speed up the repair process.
Okay, now we're going to talk about specific body defense immunity, which is also called adaptive immunity because it attacks specific viruses or pathogens. There are three characteristics to an immune response. One, it has to have antigen specificity, meaning that the immune system has to attack against specific antigens. Two, it has to be systemic or active throughout the entire body. And the third, it has to have memory. So after the initial response, subsequent responses are faster and stronger. There are two different immune responses, humoral and cellular. Humoral, with humor being the root word, which means fluid, is a type of immune response where antibodies direct the attack. Cellular response are cell-mediated, meaning that T cells are leading the attack. To stimulate the specific response, you need complete antigens. There are two characteristics to indicate whether it is complete or not. The first is immunogenicity, meaning that it causes a response from the immune system. The second is the reactivity, meaning that the antigen can react with the products of the immune system. Another way to stimulate an immune response is with a hapten, which is a molecule too small to cause a response by themselves. However, when they are binded to a normal protein or lipid, they can cause an immune response, and a common example of this is poison ivy. Antigenic determinants are part of the molecule that cause a response. The last way to cause a response is with self-antigens, also known as major histocompatibility complexes. There is 1 and 2. MHC1 are found on all major body cells except red blood cells. It is how normal cells show immune system what is happening inside of them. They either show normal or viral proteins on their cells. MHC2 is how the immune system cells communicate with each other and are a very important part of an immune response. Now we're going to talk about the important cells in the immune response. The first cells we're going to talk about are lymphocytes. They all originate in red bone marrow, become immunocompetent at different places depending on which type of lymphocyte they are. B cells mature in bone marrow, and T cells mature in the thymus, or as Mr. B calls it, T cell ninja training school. In the thymus, the cells must go through 2 tests and successfully pass to be let into the body and start working. The first is to make sure they positively select and hook to the right MHC in the cell. The second is to make sure they do not attack normal proteins. A very small percentage actually passes, and if they do not, they are forced into self-suicide. Once the cells become immunocompetent, they develop a specific surface receptor to interact with their specific antigens. Then they circulate throughout the lymphoid organs looking for their specific antigens. They are not activated until they interact with that specific antigen. The next type of cells are macrophages. They act as antigen-presenting cells and warn neighboring cells as a possible invader. Our secondary immune response and our immunological memory. We have 2 different types of. Next, we are going to be going over our secondary immune response and our immunological memory. We have 2 different types, active and passive. In active immunity, our bodies make the antibodies. There is natural active immunity, which occurs when you get sick and you get better by yourself. Active artificial immunity is when you get a shot or vaccine, so your body produces its own antibodies to that pathogen. There are different types of vaccines: live attenuated, dead, and toxoid. Live attenuated is the strongest and can last a lifetime. Dead vaccines are when dead pathogens are used to create immunity. You do need boosters to keep your immunity, but toxoid vaccines are the weakest immunity and need boosters all the time. For example, a tetanus shot, and you need a booster about every 7 years. Passive immunity is when your antibodies are made somewhere else besides your body. Naturally acquired passive immunity is usually when the mother's antibodies are passed to her baby through the placenta or claustra, which is the first milk a baby has from their mother. There is also artificial passive immunity, where you receive antibodies through shots or an IV that gives the antibodies to your body. Antibodies are also an important part of the immune response. The structure of antibodies is 2 heavy and 2 light chains, a constant region, which is the same for each class of antibodies, and a variable region, which is the antibody binding site, which sticks to the antigens. There are 5 classes of antibodies. The first is IgG, which is the most common antibody produced by plasma cells, and a lot are made in a secondary immune response. IgM is the first antibody produced in a primary immune response, and they are 5 stuck together. IgA is produced in the mucous membranes, found in sweat, saliva, and breast milk. IgAs are also the antibodies that give IgAs produced in the mucous membranes found in sweat, saliva, and breast milk. IgAs are also the antibodies that the mothers give to their babies. IgEs bind to basophils and mast cells and cause allergic responses. IgDs are found on the outside of B cells, and they make IgM and IgGs. Since each antibody comes from genetic code, there are mechanisms for their diversity. Somatic recombination and hypervariable regions create built-in recipes for different types of antibodies. There are also targets and functions. Mr. B talked about complement fixation, inactivation, neutralization, and cross-linking. Antibodies start the complement system with the antigen-antibody complex through the classical pathway. Neutralization is when the antibody binds to a site on the virus and blocks it so they cannot bind to normal body cell receptors. Then the antigen-antibody complex is formed and then they phagocytize it. Cross-linking is when an antibody grabs 2 or more antigens at a time. IgM is the best at cross-linking because there's 5 antibodies stuck together. Monoclonal labs are a test tube full of one type of antibody. Hybridomas are a mix from antibody and cancer cell. Since cancer cells live forever, you always have access to the antibodies. Cytokines are a chemical signal used in immune responses. Monokines come from macrophages. An example is interleukin-1, which causes inflammation and fever, and then lymphokines derive from T cells. An example is interleukin-2, which is the B cell and T cell dividing differentiation signal from the helper T cells. Okay, so now we're going to talk about specific body defenses immunity, which is also called adaptive immunity, because it attacks specific viruses or pathogens. There are 3 characteristics to an immune response. First, it has to have antigen specificity, meaning that the immune system has to attack against specific antigens. Second, it has to be systemic or active throughout the entire body. And third, it has to have memory. After the initial response, subsequent responses are faster and stronger. There are 2 different immune responses, humoral and cellular. Humoral, with humor being the root word, which means fluid, is a type of immune response where antibodies direct the attack. Cellular response are cell-mediated, meaning that T cells are leading the attack. To stimulate the specific response, you need complete antigens. There are two characteristics to indicate whether it is complete or not. The first is immunogenicity. There are two characteristics to indicate whether it is complete or not. The first is that it has to cause a response from the immune system. The second is reactivity, meaning that the antigen can react with the products of the immune system. Another way to stimulate an immune response are with haptens, which is a molecule too small to cause a response themselves. However, when they are binded to a normal protein or lipid, they can cause an immune response. A common example of this is poison ivy. Antigenic determinants are part of the molecule that cause a response. The last way to cause a response is with self-antigens, also known as major histocompatibility complexes. There's 1 and 2. MHC1 are found in all major body cells, except red blood cells. This all normal cells show the immune system what is happening inside of them. They either show normal or viral proteins on their cells. MHC2 is how the immune system cells communicate with each other and are a very important part of an immune response. The first cells we are going to talk about are lymphocytes. They all originate in red bone marrow but become immunocompetent in different places depending on which type of lymphocyte. B cells mature in bone marrow, and T cells mature in the thymus, or as Mr. B calls it, T cell immunity training school. In the thymus, the cells must go through 2 tests and successfully pass them to be let into the body and start working. The first is to make sure they positively select and hook to the right MHC in the cell. The second is to make sure they do not attack normal proteins. A very small percentage actually passes, and if they do not, they are forced into self-suicide. Once the cells become immunocompetent, they develop a specific surface receptor to interact with their specific antigens. Then they circulate throughout the lymphatic organs looking for their specific antigens. They are not activated until they interact with that specific antigen. The next type of cells are macrophages. They act as antigen-presenting cells and warn neighboring cells of possible invaders. Now we're going to go over our secondary immune response and our immunological memory. We have 2 different types, active and passive. In active immunity, our bodies make the antibodies. There is natural active immunity, which occurs when you get sick and get better all by yourself. Active artificial immunity is when you get a shot or vaccine, so your body produces its own antibodies to that pathogen. There are different types of vaccines: live attenuated, dead, and toxoid. Live attenuated is the strongest and can last a lifetime. Dead vaccines are when dead pathogens are used to create immunity. You do need boosters to keep your immunity, and as well as with toxoid vaccines. They are actually the weakest immunity and need boosters all the time. For example, tetanus shots, which need boosters every 7 years. Passive immunity is when your antibodies are made somewhere else besides your body. Naturally acquired passive immunity is usually when the mother's antibodies are passed through her baby through the placenta or claustra, which is the first milk a baby has from their mother. There is also artificial passive immunity, where you receive antibodies through shots or IVs. Antibodies are also an important part of the immune system. The structure of antibodies is 2 heavy and 2 light chains, a constant region, which is the same for each class of antibody, and a variable region, which is the antibody binding site, which sticks to the antigens. There are 5 classes of antibodies that we go over. The first is IgG, which is the most common antibody produced by plasma cells, and a lot are made in the secondary immune response. IgM is the first antibody produced in a primary immune response. There are 5 stuck together. IgA is produced in the mucous membranes. It is found in sweat, saliva, and breast milk. IgAs are also the antibodies that the mothers give to their babies. IgEs bind to basophils and mast cells and cause allergic responses, and IgDs are found on the outside of B cells. They make IgM and then IgGs. Since each antibody comes from genetic code, there are mechanisms for their diversity. The ones we go over in class are somatic recombination and hypervariable regions. And basically what we need to know is that they create built-in recipes for different types of antibodies. There are targets and functions of antibodies. Mr. B talks about complement fixation and activation, neutralization, and cross-linking. Antibodies start the complement system with the antigen-antibody complex through the classical pathway. Neutralization is when the antibody binds to a site on the virus and blocks it so they cannot bind to normal body cell receptors. The antigen-antibody complex is formed, and then they phagocytize it. Cross-linking is when an antibody grabs 2 or more antigens at a time. IgM is the best at cross-linking because it has 5 antibodies stuck together. Monoclonal labs are a test tube full of one type of antibody. Hybridomas are a mix between antibody and cancer cells. Since cancer cells live forever, you always have access to the antibodies. Cytokines are a chemical signal used in immune responses. Monokines come from macrophages. An example is interleukin-1, which causes inflammation and fever. Lymphokines derive from T cells. An example is interleukin-2, which is the B cell and T cell divide and differentiation signal from the helper T cells.
Next, we are going to be talking about roles of the T cells. So, helper T cells are the most important in the adaptive immune response. They help activate B cells to secrete antibodies and macrophages to destroy ingested microbes. They also help activate cytotoxic T cells to help kill infected and targeted cells. Helper T cells can only function when activated to become effector cells. They are active on the surface of antigen-presenting cells which mature during the innate immune response. Next, we're going to be talking about cytotoxic T cells, which you could also call them CD8 cells. These hunt down and destroy, so they're going to attack other cells by binding to them and releasing perforin, which is going to poke holes in the cells. Next, we're going to be talking about suppressor T cells, which are going to turn down immune response or suppress or stop. Next, we're going to be talking about organ transplants. So there are going to be 4 types of organ transplants. The first is an autograft. So this is a graft when the donor is the skin recipient. So this will be a skin graft. An example would be a burn victim. You would take skin from another place of your body that isn't affected and go ahead and transplant it on the burned area. The second is going to be an isograft. Now this is going to be a graft between identical twins. The third is an allograft. So this is a graft between humans to humans. So any type, human to human contact. The fourth is a xenograft. So now this is a graft between different species. So an example of this would be a pig valve transplant in a open heart surgery. So the pig will be donating to the human. The last topic we're going to talk about is homeostatic imbalances of immunity. So these are going to be your hypersensitivities. It is important to know the type and also the name of the sensitivity. So type 1 is immediate. So this is going to begin within seconds. And an example of this would be anaphylactic. So this is going to be in a peanut allergy. The basophils are going to release histamine. The second type is a subacute, and it is cytotoxic. So this is going to occur when you mismatch a blood transfusion. Now this will take a couple hours to show. The third type is the immune complex. So this is going to be, an example would be lupus. So lupus is caused by too much of an antigen-antibody complex. So what happens is the neutrophils actually blow up, and the guts of the neutrophils is what makes you sick. So the fourth and final type of the hypersensitivities are the delayed. So this is going to be contact dermatitis, and that is also known as poison ivy. So it'll take a few, maybe a couple days to see this, but there's also a positive function. So this is going to be a TB test. So most of us are going to be going into the medical field. You'll need a TB test to get done. So you go into your doctor's office. They'll give you a little shot. It usually goes on your forearm. And in 3 days, you go back and it'll be a little bump, and then it'll be positive or negative. So that concludes the homeostatic hypersensitivity.
Okay, so this is the drawing that Mr. B does to explain humoral and cell-mediated immunities. So these are the pathogens. This is after they get into the body past the surface barrier membranes. So now I'm drawing a macrophage. A macrophage is going to meet up with the pathogen and digest it. So once the macrophage is digested, it goes through phagocytosis. And then it's going to break up the bacteria up into pieces. So that is what I'm illustrating now. So after it phagocytoses it, and it breaks the bacteria up into pieces. MHC2, which is major histocompatibility complex, is carried outside of the cell, and it is known as an antigen presenting cell. So it is a key to start adaptive immunity. It's basically showing the other cells around it, like, hey, look at this invader, come attack me. So the antigen presenting cells meets up with the helper T cell, and they are actually not activated until they see an antigen. The helper T cell will divide and differentiate into memory cells, and it makes a bunch of the T helper cells that will recognize the bacteria, and then it will also split into T helper cells that are active. So basically, this is just from the bacteria floating around. It's pathogens outside of the cell. So this bacteria will stick to antibodies or a B cell, and then the active T helper cells will give the B cells interleukin-2, and that tells the B cells to divide and differentiate into either B memory cells or plasma cells. And those plasma cells will produce a lot of antibodies to kill that antigen. That is why it's called humoral immunity, because plasma cells are fluid, and that's what creates the antibodies to kill that type of pathogen. So now we are going to go over cell-mediated immunity. So basically, right here, these are normal body cells, and basically they have MHC1s on everything except red blood cells in the body. And MHC1s basically show normal or viral proteins on a cell. So when you have a viral protein, it will show it outside of it, and it's basically telling it, like, help me, I need help, come kill this antigen. The antigen presenting cell will show the cytotoxic T cell the pathogen that it needs to attack, but cytotoxic T cells cannot work without a T helper cell. So a T helper cell will come and give interleukin-2, and then that will divide into cytotoxic memory cells and the active cells. Then the cytotoxic T cells will go attack the viruses in the normal body cells, and that is cell-mediated immunity.